Every single one of us started as a microscopic amorphous clump of cells. I mean, there was no central nervous system to send electrical commands, right? No brain to direct traffic. Yet somehow those rapidly dividing cells knew exactly how to coordinate their movements.
Right. They just fold themselves into these incredibly complex geometric patterns.
Exactly. They build a beating heart, a breathing lung, a functioning spinal cord. If you just drop a pile of bricks, they don't spontaneously assemble into a cathedral.
Yeah, but in biology, the blueprint somehow builds the house.
And today we're doing a deep dive into how scientists are finally decoding that exact blueprint. We're looking at this groundbreaking spatial transcriptomics framework called Spateo. It's detailed in a massive paper published in the journal Cell by Q and colleagues.
And this is huge because we are essentially moving from a static, 2D understanding of cellular biology into a fully dynamic 4D molecular hologram.
A molecular hologram. I love that. So let's start with the observation problem, because historically, developmental biologists have had a really hard time actually seeing this happen, right?
Oh, absolutely. I mean, a massive problem. To understand which genes are driving the creation of an organ, you have to sequence the RNA. But the traditional method, single-cell RNA sequencing it, actually requires dissolving the tissue.
So you basically have to destroy the thing you want to study.
Exactly. It's like blending a smoothie. You get this brilliant, high-resolution readout of every gene that was active, but you completely lose the spatial context. You know you have the materials for, say, a roof and a foundation, but you have no idea where they were located.
Which brings us to spatial transcriptomics. That's the alternative, where you capture the RNA transcripts directly on a slide, right? Like a microscopic grid of DNA barcodes.
Right, exactly. When you sequence the tissue that way, the genetic data is physically tagged with an XY coordinate, which is great, but the limitation there is that it's just a flat 2D slice.
And an embryo is obviously a 3D volume. So the researchers here mapped entire mouse embryos at developmental days E9.5 and E11.5, and we are talking about tracking over 8 million individual cells.
Yeah, over 8 million. So to get that 3D volume, they had to slice those embryos incredibly thin, capture the 2D spatial data for dozens of consecutive slices, and then computationally restack them.
Which introduces what we're calling the sliced bread problem. Because slicing biological tissue is, well, it's a violent act at the microscopic scale.
Oh, totally. The knife squishes the cells, the tissue stretches, it tears, and it warps as it's mounted on the slide.
So it's literally like trying to perfectly reassemble a sliced loaf of bread where the slices have been squished or torn. If you simply tell a computer program to stack slice number 2 on top of slice number 1, nothing is going to line up.
Right, the biological landmarks just won't align. A developing blood vessel in one slice might be physically distorted 3 millimeters to the left compared to where it should be.
So I'm guessing standard image alignment software just completely falls apart here.
Yeah, it does. If you use a rigid algorithmic transformation, like just rotating or shifting the entire slice to find a best fit, you aren't actually correcting the localized warping. You're just moving the distortion around.
So to fix a stretched region of tissue without ruining the perfectly fine tissue right next to it, the algorithm needs to behave more like a selective elastic grid.
Exactly. And the Spateo framework handles this by employing non-rigid alignment driven by Gaussian processes.
Okay, Gaussian processes. Break that down for us.
So rather than applying a single mathematical transformation to the whole image, a Gaussian process models the probability of deformation across the entire surface. It analyzes the spatial coordinates of gene expression patterns, looking for identical biological landmarks between 2 adjacent slices.
Oh, so it's looking for the exact same cluster of specific neurons in both slices.
Right, and then it calculates a customized vector field of deformations. So it identifies that, say, a cluster of neurons is slightly stretched in slice 2, and it mathematically pulls just that localized region back into its proper biological shape,
matching it to the footprint in slice 1. But wait, doesn't that introduce a massive risk of error propagation?
What do you mean?
Well, if slice 2 is aligned to slice 1 and slice 3 is aligned to slice 2, by the time you reach slice 50, a microscopic calculation error at the very beginning has totally snowballed, right? You could end up with an embryo that twists like a corkscrew.
Oh, yeah, structural drift. That's a huge issue. To prevent that, Spateo implements multi-slice refinement. It doesn't rely solely on pairwise alignment.
Okay, so what does it do instead?
It anchors the calculation by analyzing a rolling neighborhood of slices, usually like 3 slices forward and 3 slices back. By jointly modeling the spatial transcriptomics of an entire block of tissue simultaneously, the algorithm creates a sort of moving average of structural integrity.
Ah, so it prevents those tiny localized adjustments from accumulating into a macro-level distortion. That solves the stretching and tearing. But the sources mention another, much more insidious geometric illusion that happens when you stack these 2D cross-sections.
Ah, yes, the Z-shift problem. Or, as it's sometimes called, the banana problem.
The banana problem. I love that.
Huh.
So if you take a biologically curved object, slice it into perfectly flat circles, and ask an algorithm to align the center math of those circles, the computer is just going to stack them perfectly straight up, right?
Exactly. The algorithm inherently seeks the path of least resistance for alignment, which is a straight cylinder. So the overarching natural curve of the organ is completely mathematically erased.
But a developing spinal cord, for example, has this distinct, sweeping biological curve. So how do they get that back?
To rescue that macrogeometry, Spateo introduces mesh correction. Before the embryo is ever sliced for transcriptomics, the researchers actually generate a 3D morphological mesh, basically an intact whole embryo 3D scan.
Oh, so they use the unbroken external topology as like a bounding box.
You got it. They force the computationally aligned stack of slices to conform to that overarching 3D mesh, so the Gaussian processes handle the microscopic localized tissue repair, while the mesh correction forces the macrostructure to snap back into its natural curve.
That is incredibly clever. So, quick recap for you listening. Imagine trying to rebuild a 3D map of your own neighborhood using only shredded photographs. That is the scale of what Spateo just achieved. They repaired the microscopic tears, anchored the alignment to prevent drift, and snapped it all into a perfect 8 million cell 3D volume.
Right, but having a flawless 3D model is entirely useless if you don't know how to navigate it.
Exactly, because if I drop you inside the curving organic tube of a developing neural tube, standard Cartesian coordinates like your standard X, Y, and Z axes are going to fail immediately.
They really do. Cartesian coordinates work beautifully for rigid, engineered structures, but biological organs are these squiggly, emergent shapes.
Right. If you want to measure the diffusion of a morphogen from the inner lining of a curved tube to the outer edge, a straight Cartesian vector will likely just cut right through the empty space in the middle of the curve.
Or it might even exit the organ entirely and re-enter it further down. It fundamentally misunderstands the biological territory.
So how does Spateo navigate this?
It uses a 3D digitization module based on Poisson's equation.
Wait, really? Poisson's equation, isn't that typically used in astrophysics to map the gravitational pull of planets? Or in electromagnetism for electrical fields?
It is, yeah. It's a brilliant cross-disciplinary leap. Applying physics-based field equations to a non-uniform biological medium implies they are treating the organ boundaries like the terminals of a battery.
Oh, wow. Why use physics math for biology? Give me an example of how that actually works in practice.
Let's look at how they applied this to the ZLI, the zona limitans intrathalamica. It's a highly complex 3D signaling center in the developing forebrain. It acts as a primary organizer, basically sending out genetic signals that tell surrounding cells whether to become the anterior or posterior parts of the thalamus.
Okay, so understanding how gene expression changes as you move away from the ZLI is crucial.
Exactly. So to map that, the framework identifies the geometric boundaries of the ZLI and assigns them contrasting numerical values. The inner boundary acts as a source, set to a value of +1, and the outer boundary acts as a sink, set to -1.
And then, by solving Poisson's equation across the tissue between those two boundaries, the algorithm generates a contour-hugging map of equipotential lines.
Right. Think of a topographic map of a mountain range. The contour lines follow the exact winding shape of the peaks and valleys. By calculating the potential field inside the organ, Spateo basically creates a biological topographic map.
So instead of telling the software to look 3 millimeters along the x-axis, researchers can instruct it to track the biological gradient along a specific equipotential line.
Exactly. And this reveals the hidden architecture of organogenesis. Using these custom curd-hugging spatial coordinates, the researchers were able to cleanly map the opposing gradients of specific genes like SLIT2 and DBS1 across the developing spinal cord.
And those gradients dictate exactly how the progenitor cells in the spinal cord mature and differentiate into specialized neurons, right?
They do. It proves that the physical geometry of the organ is inextricably linked to how the genetic code is executed.
That is just fascinating. So for everyone following along, think about how temperature drops as you climb a mountain. Spateo is basically mapping the temperature drops of gene expression across the squiggly curves of an organ.
That's a perfect way to visualize it.
Okay, so we have the structure repaired, and we have an accurate contour-hugging coordinate system. But the cells within this 3D space, they aren't just static elements on a map. They're constantly communicating.
Oh, yeah. Life isn't silent. And the sources dedicate a massive portion of the framework to calculating this cell-cell interaction, or CCI.
So how do we figure out how these cells are gossiping across this 3D space?
Spateo uses a spatially weighted generalized regression model to map the entire molecular signaling cascade. It separates the short-range whispers from the long-range shouting.
Right, short-range signaling involves membrane-bound ligands. These are proteins anchored to the surface of a cell that require physical membrane-to-membrane contact with a neighboring cell to trigger a reaction.
And then long-range signaling involves secreted molecules that a cell basically pumps out into the extracellular matrix, which then diffuse across those equipotential fields we just discussed to reach distant targets.
And what blows my mind is that the framework doesn't just register that two cells are near each other. It traces the full biological axis. It identifies the ligand being produced by cell A.
Yeah, and it maps that ligand traversing the spatial gap to bind with a highly specific receptor on the surface of cell B.
Right, which then calculates how that receptor activation triggers an intracellular transcription factor, which physically enters the nucleus of cell B to turn on a target gene. It tracks the physical cause and the genetic effect across the entire 3D volume.
The depth of that interaction mapping is completely unprecedented. But computing that chain of events for every single cell introduces a catastrophic mathematical hurdle.
I mean, yeah, it's an insurmountable scale problem. If we have 8 million cells and thousands of genes, calculating every single interaction for every single cell must require a supercomputer the size of a building.
The combinatorial explosion would crash any server farm, honestly.
So how is this mathematically possible? They have to be using some form of dimensionality reduction, right? Something akin to a downsampling algorithm in image processing, where you reduce the file size by grouping similar pixels, but applying to biological signaling.
You are hitting on exactly how they solved it. Spateo uses a computational strategy called stratified subsampling.
Stratified subsampling. Okay, how does that work?
The algorithm analyzes the spatial transcriptomic data and groups the cells into localized transcriptionally similar clusters. So if you have a cluster of 100 cells packed tightly together in the developing midbrain, expressing the same baseline genes, the algorithm just selects a single representative exemplar cell.
So instead of calculating every single cell, it picks an exemplar. It performs that massive, computationally heavy regression modeling to calculate the complex chemical signaling environment for just that one cell.
Exactly. And because the surrounding 99 cells are sitting in the exact same microenvironment, bathed in the same secreted morphogens, it propagates that highly detailed signaling profile out to the immediate localized neighborhood.
It's an incredibly clever computational shortcut. It drastically reduces the computational matrix while preserving the high-resolution signaling map.
It really is. By analyzing these subsampled interactions, researchers can identify precisely which chemical signals dictate cell fate in different regions of the embryo. They can observe how a wave of secreted signaling molecules physically washes over a region of unspecialized tissue, triggering the transcription factors that irreversibly commit those cells to becoming, say, cardiac muscle.
So if you could eavesdrop on the chemical whispers of your own cells, what do you think they'd be saying to each other? That's what this tool lets us do. But up until this point, we've essentially been analyzing a frozen, highly detailed communicative snapshot.
Right, and organogenesis is inherently a dynamic moving process. Tissues fold, they migrate, they expand and contract. The embryo at day E9.5 looks vastly different than the embryo at E11.5.
Right. So to truly understand how the blueprint builds the house, the framework has to integrate the fourth dimension, time.
This is where Spateo introduces morphometric vector fields. The framework maps the cellular coordinates from the earlier developmental time point to the later one to track their physical migration.
And to do that, they borrow concepts directly from differential geometry and fluid dynamics, right?
They do. They calculate metrics like divergence. So is a specific region of tissue physically expanding outward, like a high-pressure system, or is it compacting inward? And they measure curl, which is rotation is a specific group of cells twisting around a central axis, along with acceleration and torsion.
It literally treats the developing embryo like a 3D weather map. We're looking at the wind currents, the twisting tornadoes of tissue, and the high-pressure zones of migrating cells.
Exactly like a weather map.
But let's trace the causality here, because this is where the physics actually answers the biological mystery. Why does calculating the geometric curl of a tissue matter to a developmental geneticist? How does identifying a microscopic tissue tornado lead to a genetic discovery?
Let's look at the asymmetric development of the mouse heart, which is one of the earliest and most complex organs to form. To create the separate chambers of the heart, the lineal heart tube has to undergo a highly specific asymmetrical looping process. It must twist perfectly to the right.
And if it loops the wrong way or fails to loop, the embryo develops severe congenital heart defects.
Right. So by applying morphometric vector fields to the 3D data, the algorithm can mathematically isolate the exact spatial coordinates of the tissue undergoing high torsion. It filters out the millions of static cells and leaves you with only the specific cells that are actively driving that physical twist.
Oh, wow. So once you have isolated those rotating cells, you just query their transcriptomic data. You look at their RNA.
Exactly. And by doing this, the researchers were able to pinpoint the exact morphogenic drivers, specific transcription factors like TBX2 and PIX2 that are highly expressed in the twisting region.
Wait, PIX2 is heavily involved in establishing left-right asymmetry in vertebrates. So the framework allows scientists to use the macroscopic physical movement of the tissue as the direct filter to find the microscopic genetic instructions driving that exact movement.
Yes, the mechanical rotation is a direct readout of the localized gene expression, and they actually applied the same vector field analysis to the morphogenesis of the Drosophila gut, the fruit fly midgut, and were able to similarly map the genetic drivers of its complex folding patterns.
So we are essentially watching the choreography of life. It's biology visualized through the lens of fluid dynamics. We are moving entirely away from a reductionist view of biology.
We really are. We are no longer looking at single cells in an isolated dish and guessing what they do. We are observing the true tissue ecology, analyzing the mechanical forces, the chemical signaling, and the genetic execution all simultaneously.
It is a profound integration of disciplines. We started with dead, flattened, disconnected slices of biological tissue. We utilize the probabilistic math of Gaussian processes to computationally repair the microscopic tearing and whole embryo mesh correction to snap the stacked volume back into its natural curve.
Right, and then we deployed astrophysics field equations to generate custom contour-hugging coordinates inside those organic curves.
We eavesdropped on the cellular gossip, using stratified subsampling to efficiently map the massive cellular signaling network, tracing the message from membrane ligand down to nuclear DNA.
And finally, we applied the fluid dynamics of morphometric vector fields to watch those cells physically migrate and twist to build an asymmetric heart.
And crucially, the authors of this paper didn't just publish the math and lock it away. They built an interactive software tool called Spateo-viewer, which allows researchers anywhere in the world to boot up these 3D models. They literally call it the Google Earth of spatial transcriptomics.
Yeah, you can just zoom in on a developing midbrain, rotate it, and track the equipotential lines of gene expression yourself. It democratizes a level of developmental insight that was essentially science fiction a decade ago.
Which leaves us with a fascinating, forward-looking reality to consider. Because if a computational framework can now map, digitize, and track exactly how the physical forces and chemical signals guide unspecialized cells into building a functioning organ.
We are looking at the foundational code for tissue engineering.
Exactly. If we understand the precise sequence of vector fields and morphogen gradients required to loop a heart tube, how long until we can use these molecular holograms to completely digitally simulate the growth of artificial human organs in a lab?
Or even run a digital twin simulation of an embryo to predict a congenital defect based on its early signaling environment.
Allowing us to understand exactly where the assembly line breaks down before it happens physically. We've spent decades trying to read the biological blueprint. Today, we are finally watching the house build itself. Thank you for joining us on this deep dive into the source material. We'll see you next time.